Hello, everyone. Today we're diving into the fascinating world of Sporozoa. These are some of the most important microscopic organisms you've probably never heard of. So, what exactly are Sporozoa? Well, Sporozoa is an older scientific term used to describe a group of parasitic protozoans. These are single celled organisms that have a very special way of reproducing through spores. Here we can see a simplified diagram of a sporozoan cell. Notice the cell membrane, the nucleus in the center, and those small red dots. Those represent the spores that these organisms use for reproduction. The key thing to understand about sporozoa is that they are parasites. Think of them as tiny invaders. They don't live freely in the environment. Instead, they live inside the cells of other organisms, which we call their hosts. When these tiny invaders reproduce, they create spores, special survival forms that can spread to new hosts. This spore formation is one of their defining characteristics and makes them incredibly successful parasites. So here's your key takeaway for this introduction. Sporozoa are parasitic, spore-forming single-celled organisms. They're microscopic invaders that live inside other organisms and reproduce through specialized spores. In our next sections, we'll explore these characteristics in much more detail. Now that we've introduced these fascinating organisms, we're ready to dive deeper into what makes them so unique and important in the world of microbiology. The story of Sporozoa begins in 1879, when German zoologist Carl Georg Friedrich Rudolf Leuckart first coined this term to describe a fascinating group of microscopic organisms. Leuckart grouped these organisms based on one key observation. Unlike other protozoans that could swim or crawl, these creatures appeared to lack any structures for movement. These sporozoans are unicellular organisms, meaning each individual is a complete living being contained within a single cell. Despite being just one cell, they carry out all the functions necessary for life. All sporozoans are parasites, living inside the cells of their host organisms. They have evolved to absorb nutrients directly from their hosts, making them completely dependent on other living beings for survival. So remember, Sporozoa are single-celled parasites that lack structures for movement and survive by absorbing nutrients from their host cells. This basic definition, established in 1879, laid the foundation for understanding these important microscopic organisms. The term Sporozoa is no longer used in modern scientific classification. Scientists have moved to a more accurate system based on the actual structures these organisms possess. Today, these organisms are classified under the phylum Apicomplexa. This name comes from a special structure they all share called the apical complex. Here we see a typical apicomplexan cell. The apical complex is located at one end of the cell and consists of several specialized structures including polar rings, a conoid, and secretory organelles. This apical complex is crucial for the parasite's survival. It acts like a molecular drill, helping the parasite penetrate and invade host cells. The secretory organelles release enzymes and proteins that break down the host cell membrane. This structural classification system is much more accurate than the old Sporozoa grouping because it's based on actual shared anatomical features rather than just the absence of movement organelles. Understanding this modern classification helps scientists better study these important parasites and develop more effective treatments and prevention strategies. The most defining characteristic of sporozoans is their parasitic nature. But they're not just any kind of parasite. They are specifically endoparasites. What does endoparasite mean? Let's compare this to other types of parasites to understand the difference. External parasites, like fleas or ticks, live on the outside of their host. They attach to the skin or fur, but don't actually enter the host's cells. Sporozoans are completely different. They are endoparasites, which means they actually penetrate into the host cells and live inside them. They literally set up shop inside the cell. This intracellular lifestyle gives sporozoans several advantages. They can hide from the host's immune system, access nutrients directly from inside the cell, and use the cell's own machinery for their reproduction. Let's see how a sporozoan actually invades a host cell. This process is quite remarkable and shows just how specialized these parasites have become. Here we see a sporozoan approaching a host cell. The sporozoan has specialized structures that help it penetrate the cell membrane. The sporozoan moves toward the cell and begins the invasion process. It uses its apical complex, a specialized structure, to penetrate the cell membrane. Once inside, the sporozoan is now protected within the host cell. It can access the cell's nutrients and begin its life cycle, all while being hidden from the host's immune defenses. This endoparasitic lifestyle is what makes sporozoans so successful as parasites, but also what makes the diseases they cause so difficult to treat. Understanding this characteristic is crucial for developing effective treatments. One of the defining characteristics of Sporozoa is their unique method of reproduction through spore formation. These microscopic organisms have evolved a highly effective reproductive strategy. The process begins with a mature sporozoan cell. Inside this parent organism, specialized reproductive processes create multiple spores through a process called sporulation. Through sporulation, a single parent cell can produce many spores. Each spore is like a tiny, protected package containing all the genetic material needed to start a new infection. Spores have three key features that make them incredibly effective for parasitic reproduction. First, they have a protective coating that helps them survive harsh environmental conditions. Second, spores can remain dormant for extended periods, waiting for the right conditions or host to become available. This dormancy allows them to survive outside a host. Third, when conditions are right, spores can quickly activate and become highly infective, allowing them to establish new infections in suitable hosts. In summary, spore formation is a crucial reproductive strategy that allows Sporozoa to survive, spread, and continue their parasitic lifestyle. These tiny packages are perfectly designed for transmission and infection. Unlike many other microorganisms, sporozoans have a unique characteristic that sets them apart from their microscopic neighbors. They lack the typical structures that most single-celled organisms use to move around. Most microorganisms have specialized structures for movement. Paramecium uses tiny hair-like cilia that beat in coordinated waves. Euglena propels itself with a whip-like flagellum. Amoeba extends pseudopodia, or false feet, to crawl along surfaces. Sporozoans are completely different. They have smooth cell surfaces with no cilia, no flagella, and no pseudopodia. Their cell membrane is unadorned with any of the typical movement structures we see in other microorganisms. So how do sporozoans get around without these movement structures? They rely on their host organisms. They use passive transport through blood circulation, are carried by host cell movement, or employ a special gliding motility that doesn't require external organelles. This lack of locomotory organelles is a defining characteristic that reflects their parasitic lifestyle. Since they live inside host cells, they don't need to swim through water or crawl across surfaces like free-living microorganisms do. Sporozoans are remarkable for their incredibly complex life cycles. Unlike simple organisms that just grow and divide, these parasites go through multiple distinct stages, each with a specific purpose. A typical sporozoan life cycle involves 6 major stages, each serving a crucial function. The cycle begins with infectious spores that seek out host cells. Once inside a host cell, the parasite begins asexual reproduction, rapidly multiplying to create many copies of itself. This allows the infection to spread throughout the host organism. At certain points in the cycle, sporozoans switch to sexual reproduction. This creates genetic diversity and produces specialized forms that can survive transmission between hosts. The sexual reproduction creates new spores with enhanced survival capabilities. These spores are designed to withstand harsh conditions and successfully infect new hosts. Most sporozoans require 2 or more different host species to complete their life cycle. This might include a vertebrate host like a human and an invertebrate vector like a mosquito. This complexity makes sporozoans incredibly successful parasites, but it also creates vulnerabilities. Breaking any part of the cycle can stop the infection, which is why understanding these life cycles is crucial for developing treatments and prevention strategies. One of the most well-known and devastating examples of a sporozoan is Plasmodium falciparum, the parasite responsible for causing malaria. Plasmodium falciparum is a single-celled parasitic organism that belongs to the Sporozoa group. This microscopic parasite has a complex internal structure typical of eukaryotic cells. This parasite causes malaria, a serious infectious disease that is transmitted to humans through the bites of infected mosquitoes. The parasite invades and destroys red blood cells, causing symptoms like high fever, chills, and severe anemia. The global impact of malaria is truly staggering. Let's look at the devastating numbers from recent data. In 2021 alone, malaria affected 247 million people worldwide. This massive number represents the scale of this parasitic disease's reach across the globe. Even more devastating is that malaria caused 619,000 deaths in that same year. Each of these numbers represents real human lives affected by this tiny sporozoan parasite. The majority of these deaths occur in sub-Saharan Africa, where the disease burden is highest and healthcare resources are often limited. Plasmodium falciparum demonstrates how a microscopic sporozoan can have enormous global health consequences, making it one of the most important parasites to understand and combat. Eimeria represents another important genus of coccidian parasites within the Apicomplexa phylum. These microscopic organisms are responsible for causing significant diseases in various animal species. Eimeria parasites exist as oocysts, which are protective structures containing infectious sporozoites. These oocysts are the form that spreads between animals and survives in the environment. Eimeria species infect a wide range of domestic animals, including cattle, poultry, sheep, rabbits, and pigs. Each animal species typically has its own specific Eimeria species that infect them. These parasites cause a disease called coccidiosis, which primarily affects the intestinal tract of infected animals. The disease can range from mild digestive upset to severe, life-threatening conditions. Coccidiosis symptoms include diarrhea, weight loss, dehydration, and reduced growth rates. In severe cases, especially in young animals, the infection can be fatal if left untreated. The economic impact of Eimeria infections is substantial. In the livestock industry, coccidiosis causes millions of dollars in losses annually through reduced productivity, treatment costs, and animal mortality. Key takeaways about Eimeria. These coccidian parasites are species-specific, cause significant economic losses in livestock, and require proper management through hygiene, medication, and vaccination programs to control their spread. Cryptosporidium represents one of the most significant waterborne parasitic threats worldwide. This microscopic organism causes a diarrheal disease called cryptosporidiosis, affecting millions of people and animals each year. Cryptosporidium exists as tiny oocysts measuring only 4 to 6 micrometers in diameter. These thick-walled structures contain 4 sporozoites, the infective stage of the parasite. Their small size and resistant outer wall make them extremely difficult to filter from water. These oocysts contaminate water sources, including drinking water, swimming pools, and recreational water bodies. Just 10 oocysts can cause infection in humans, making Cryptosporidium one of the most infectious waterborne pathogens. Cryptosporidiosis primarily affects the small intestine, causing severe watery diarrhea that can last for weeks. The disease affects both humans and animals, including cattle, sheep, and other livestock, making it a significant concern for both public health and agriculture. The main symptoms include persistent watery diarrhea, stomach cramps, dehydration, vomiting, and sometimes a low-grade fever. In healthy individuals, symptoms typically last 1 to 2 weeks, but in immunocompromised patients, the infection can be life-threatening and chronic. Cryptosporidium stands out among parasites for its remarkable resistance to standard water treatment methods, including chlorine disinfection. This makes it a persistent threat in water supplies worldwide, requiring specialized filtration and UV treatment for effective removal. Toxoplasma gondii is one of the most successful parasites on Earth. This single-celled organism is the causative agent of toxoplasmosis, a disease that affects millions of people worldwide. This crescent-shaped parasite belongs to the phylum Apicomplexa. Like other sporozoans, it has specialized structures that help it invade host cells and establish infection. What makes Toxoplasma gondii remarkable is its incredibly broad host range. It can infect virtually any warm-blooded animal, including humans, birds, and mammals. However, cats hold a special role as the definitive host. Only in cats can Toxoplasma gondii complete its full sexual reproductive cycle and produce the infectious oocysts that spread the parasite. Toxoplasma gondii is incredibly widespread and significant for several reasons. It infects approximately one-third of the world's population, though most infections are asymptomatic. However, the parasite can be dangerous for pregnant women as it can cause serious birth defects. Recent research also suggests it may influence brain function and behavior in infected individuals. Understanding Toxoplasma gondii is crucial for public health, especially given its prevalence and potential impacts on human health and behavior. Babesia represents another important example of sporozoan parasites. This genus consists of tick-borne organisms that specifically target red blood cells in their hosts. Unlike many other sporozoans, Babesia parasites are transmitted through tick bites. Infected ticks serve as vectors, transferring the parasites from one host to another during blood feeding. Once transmitted, Babesia parasites specifically target red blood cells. They invade these cells and reproduce inside them, causing the characteristic ring-shaped or pear-shaped forms visible under microscopic examination. The transmission process occurs when an infected tick attaches to a host and feeds on blood. During this feeding, Babesia sporozoites are injected into the bloodstream, where they quickly locate and invade red blood cells. Babesiosis, the disease caused by Babesia parasites, produces symptoms remarkably similar to malaria. Patients typically experience fever, chills, fatigue, and muscle aches. The destruction of red blood cells leads to hemolytic anemia and can cause dark-colored urine. This similarity to malaria symptoms often makes initial diagnosis challenging, requiring specific laboratory tests for accurate identification. To summarize, Babesia represents a significant tick-borne sporozoan that demonstrates the diversity of parasitic strategies. Its specific targeting of red blood cells and malaria-like presentation make it an important pathogen to understand in both veterinary and human medicine. Thalaria represents another important example of sporozoan parasites. This genus consists of tick-borne parasites that specifically target cattle and other livestock. Thalaria parasites are single-celled organisms that belong to the phylum Apicomplexa. Like other sporozoans, they possess an apical complex that helps them invade host cells. Thalaria parasites are transmitted exclusively through tick bites. Ticks serve as both vectors and intermediate hosts, picking up the parasites when feeding on infected cattle. When an infected tick feeds on cattle, it injects Thalaria parasites into the bloodstream. The parasites then invade red blood cells and white blood cells. Thalariosis causes serious symptoms in cattle, including fever, anemia, weight loss, and reduced milk production. In severe cases, the disease can be fatal if left untreated. The economic impact of the thalarosis on the cattle industry is substantial. The disease reduces productivity, increases treatment costs, causes cattle mortality, and leads to significant market losses for farmers and the agricultural sector. Thalaria demonstrates how sporozoan parasites can have far-reaching consequences beyond individual animal health, affecting entire agricultural economies and food security. Before modern molecular techniques revolutionized taxonomy, scientists classified organisms based on observable characteristics. The traditional classification system placed Sporozoa under the kingdom Protozoa. In this outdated system, all Sporozoa were grouped together in the kingdom Protozoa, which encompassed all single-celled eukaryotic organisms. This was a broad category that included many diverse microorganisms. This classification was based on several key observable features. Scientists grouped these organisms together because they were all single-celled, had a eukaryotic cell structure with a nucleus, lived as parasites, and formed spores. To understand this classification, imagine a typical single-celled eukaryote. Unlike bacteria, these organisms have a true nucleus containing their genetic material, along with other specialized structures called organelles. This traditional system was first proposed in 1879 and remained the standard classification for many decades. However, as our understanding of these organisms improved, scientists realized this grouping was too simplistic. The problem with this traditional classification was that it grouped together organisms that were actually quite different from each other. Modern molecular analysis revealed that many so-called Sporozoa were not closely related, despite sharing similar lifestyles and characteristics. This realization led scientists to develop new classification systems based on genetic relationships rather than just physical similarities. The traditional Sporozoa classification became outdated as more accurate methods emerged. The modern classification system places these organisms in a much more accurate framework based on evolutionary relationships and cellular characteristics. Today, these organisms are classified in the kingdom Protista. This kingdom includes all single-celled eukaryotic organisms that don't fit into the plant, animal, or fungal kingdoms. Within kingdom Protista, these organisms belong to phylum Apcomplexa. This phylum is defined by the presence of a unique cellular structure called the apical complex. The apical complex is a specialized organelle found at one end of the cell. It contains structures that help the parasite penetrate and invade host cells. This modern classification reflects a much more accurate understanding of evolutionary relationships. It groups organisms based on shared characteristics and common ancestry, rather than just superficial similarities. The kingdom Protista and phylum Apicomplexa classification system provides scientists with a framework that better reflects the true nature and relationships of these important parasitic organisms. This modern understanding helps researchers better study these organisms and develop more effective treatments for the diseases they cause. Sporozoans are renowned for having some of the most complex life cycles in the biological world. Understanding this complexity is crucial to comprehending how these parasites survive and spread. The hallmark of sporozoan life cycles is the alternation between two distinct types of reproduction, sexual and asexual. This alternation allows them to maximize both genetic diversity and population growth. Sexual reproduction creates genetic variation, helping parasites adapt to host defenses and environmental changes. Asexual reproduction allows rapid multiplication when conditions are favorable. Even more remarkable is that these complex life cycles typically require multiple hosts to complete. Most sporozoans need at least 2 different host species. The life cycle typically involves a vertebrate host, such as a human or other mammal, and an invertebrate host, often an insect vector like a mosquito or tick. The parasite must successfully transfer between these hosts to complete its life cycle. Different stages of reproduction often occur in different hosts, making the cycle highly dependent on both host species. This multi-host requirement makes sporozoan life cycles incredibly complex, but also creates vulnerabilities. Breaking the cycle at any point can prevent transmission and disease. Apicomplexan parasites have a unique spore morphology that sets them apart from other parasitic organisms. Their spores are called oocysts, and understanding their structure is crucial for comprehending how these parasites survive and spread. The apicomplexan spore, called an oocyst, has a distinctive structure. It consists of a tough, protective outer wall that encases multiple sporozoites inside. This wall is incredibly resilient and allows the oocyst to survive harsh environmental conditions. Each sporozoite within the oocyst is a highly specialized infective form. At its front end is the apical complex, a unique organelle that gives the Apicomplexa phylum its name. This complex contains specialized structures that help the sporozoite penetrate host cells. The oocyst's tough wall makes it incredibly resistant to environmental stresses. It can survive exposure to sunlight, temperature extremes, moisture changes, and other harsh conditions that would destroy most other microorganisms. This durability is key to the parasite's transmission strategy. When conditions are right, the oocyst becomes the infective stage. Upon reaching a suitable host, the oocyst releases its sporozoites, which then use their apical complexes to penetrate host cells and begin the infection process. This completes the transmission cycle of apicomplexan parasites. Understanding apicomplexan spore morphology helps us appreciate how these parasites have evolved sophisticated survival and transmission strategies, making them some of the most successful parasites in nature. Recent years have brought exciting developments in vaccine research against sporozoan diseases. Scientists are making significant progress in creating effective vaccines, particularly against malaria. Research laboratories around the world are working intensively to develop vaccines against sporozoan parasites. This represents one of the most promising approaches to combat these deadly diseases. The most promising approach uses sporozoites, which are the infective stage of the parasite transmitted by mosquitoes. These sporozoite-based vaccines have shown remarkable results in clinical trials. Clinical trials have demonstrated impressive efficacy rates for these sporozoite vaccines. Let me show you the remarkable results that researchers have achieved. Clinical trials have shown that sporozoite vaccines can achieve efficacy rates of 85% or higher. These vaccines provide protection for 18 to 19 months without requiring booster shots, and they have proven to be safe and well tolerated by patients. These vaccine developments represent a major breakthrough in the fight against sporozoan diseases. The high efficacy rates and long-lasting protection offer hope for controlling malaria and other deadly parasitic infections that affect millions of people worldwide. This progress in vaccine development marks a significant step forward in our ongoing battle against sporozoan parasites and the diseases they cause. First-generation PFSPZ vaccines represent a major breakthrough in malaria prevention. These vaccines use whole Plasmodium falciparum sporozoites to provide protection against malaria infection. PFSPZ stands for Plasmodium falciparum sporozoite. These vaccines contain whole, live sporozoites that have been specially treated to create immunity without causing disease. Clinical trials have demonstrated that first-generation PFSPZ vaccines are remarkably safe and well tolerated. Participants experienced no serious adverse effects, making these vaccines suitable for widespread use. These vaccines have proven highly effective against controlled human malaria infection, or CHMI. In clinical trials, vaccinated participants showed strong protection when deliberately exposed to malaria parasites under controlled conditions. One of the most impressive features of these vaccines is their long-lasting protection. A single vaccination provides immunity for 18 to 19 months without requiring any booster shots. These promising results come from clinical trials conducted in Africa, where malaria is endemic and poses the greatest threat. Testing in real-world conditions where malaria is common demonstrates the vaccine's practical effectiveness. First-generation PFSPZ vaccines represent a significant advancement in malaria prevention, offering safe, long-lasting protection that could transform malaria control efforts in endemic regions. Drug resistance in protozoan parasites has become one of the most pressing challenges in modern medicine. These microscopic organisms are constantly evolving to survive our best treatments. Initially, when we develop a new drug, it works effectively against the target parasites. The drug molecules successfully attack and eliminate the majority of the parasite population. However, some parasites survive the initial treatment. These survivors carry genetic mutations that make them less susceptible to the drug. This is where the problem begins. These resistant parasites then reproduce rapidly, passing their resistance genes to their offspring. Over time, the entire population becomes dominated by drug-resistant strains. This creates a serious medical challenge. The same drugs that once worked effectively are now useless against these evolved parasites. Patients who could previously be cured now face treatment failure. The solution requires continuous development of new drugs and treatment strategies. Scientists must stay one step ahead of the parasites, developing novel compounds that can overcome existing resistance mechanisms. Drug resistance is an evolutionary arms race. As parasites adapt to our treatments, we must continuously innovate to stay ahead. This ongoing challenge requires sustained research investment and new approaches to combat these resilient microscopic enemies. Advances in diagnostic tools have revolutionized how we detect protozoan infections. Early and accurate diagnosis is absolutely crucial for effective treatment and disease control. Traditional diagnostic methods relied heavily on microscopic examination of blood smears and tissue samples. While effective, these methods were time-consuming and required highly skilled technicians. Modern diagnostic tools have transformed this landscape. Molecular techniques like PCR can detect parasites in minutes rather than hours, with much higher sensitivity and specificity. Let's examine specific diagnostic advances that have made the biggest impact in detecting sporozoan parasites. Polymerase chain reaction, or PCR, can amplify tiny amounts of parasite DNA, making detection possible even when parasite levels are extremely low in the blood. Rapid diagnostic tests provide results in just 15 to 20 minutes using simple blood samples. These are especially valuable in remote areas where laboratory facilities may not be available. These diagnostic advances have three major benefits. Faster results lead to quicker treatment, higher accuracy reduces misdiagnosis, and portable tests make diagnosis possible anywhere. These diagnostic improvements are saving lives by enabling earlier detection and treatment of sporozoan infections, particularly in areas where these diseases are most prevalent. Experts in parasitology and disease control have reached an important consensus. No single method alone can effectively control complex parasitic diseases like theileriosis and other tick-borne illnesses. Instead, experts emphasize that integrated control represents the most effective strategy. This comprehensive approach combines multiple complementary methods to create a robust defense against parasitic diseases. The integrated control approach consists of several key components working together. First, immunization and vaccination programs provide the foundation by building host immunity against parasitic infections. Second, vector control targets the organisms that transmit parasites, such as ticks and mosquitoes. This includes environmental management and targeted treatments to reduce vector populations. Third, surveillance and monitoring systems provide early detection of outbreaks and track the effectiveness of control measures. This allows for rapid response and adaptive management strategies. Fourth, environmental management focuses on modifying habitats to reduce favorable conditions for parasites and their vectors. This includes pasture management and reducing breeding sites. Finally, treatment and therapeutic interventions provide direct medical care for infected individuals while also reducing the reservoir of infection in the population. Expert consensus emphasizes that these components must work synergistically. When implemented together, they create multiple barriers against parasitic diseases, making it much harder for parasites to establish and maintain infections. This integrated approach is particularly crucial for tick-borne diseases like theileriosis, where the complex life cycles of parasites and their vectors require multiple intervention points to achieve effective long-term control. Understanding Sporozoa, now classified as apicomplexans, is absolutely crucial for addressing some of the world's most devastating parasitic diseases. These microscopic parasites have a massive global impact. Malaria alone affects 247 million people worldwide, causing over 619,000 deaths annually, primarily in sub-Saharan Africa. Beyond the human cost, these parasitic diseases create billions of dollars in economic losses through healthcare costs, lost productivity, and reduced quality of life. From malaria and cryptosporidiosis to toxoplasmosis and babesiosis, apicomplexan parasites cause a wide range of serious infections that affect both humans and animals worldwide. The good news is that continued research is making real progress. Scientists are developing new vaccines, creating advanced diagnostic tools, and discovering novel treatment strategies to combat these infections. By understanding the biology, life cycles, and characteristics of these parasites, we can develop better ways to prevent, diagnose, and treat the diseases they cause. Knowledge is our most powerful weapon against these ancient enemies. Together, through continued research and global cooperation, we can combat these infections and protect the health of people and animals around the world. Thank you for joining us on this journey through the fascinating and important world of Sporozoa. Your understanding of these organisms contributes to our collective fight against parasitic diseases.